Welcome to Alpha Genomics seminars and tutorial series. We are going to discuss about primer designing and use of primers in PCRs. So what is PCR, what are the reagents which are needed for PCR, and specifically talking about the primers, the role of primers in PCR and designing the primers for PCR reaction. We'll talk about this. In this talk we will cover some basic topics like some terminology which is pertinent to PCR, choosing appropriate primers either from the literature or de novo designing for the primers, general characteristics of good primers, and the PCR conditions that can affect the annealing of primers during the PCR reaction, differences in primers between the types of PCRs like the conventional PCRs, real-time PCR, next generation sequencing, any influence of primer design and choice that will be on the results or success of the PCR reaction, some logistics topics such as the ordering primers from different companies, storing the primers and recomposing the primers, troubleshooting such as the organisms where pairing may be lesser strong, and software to design the primers. And at the end we will try to use some software for primer designing as well. So the basic terminology about the PCR reaction. What is the PCR? PCR is polymerase chain reaction, in which we are going to amplify a particular piece of DNA from the whole genome, and the objective of this amplification is to use that for either for Sanger sequencing or only for gel electrophoresis for absence of bands, and so on. For a PCR or polymerase chain reaction, we need to have a pair of primers which are conventionally called as forward primer and reverse primer. These primers are the places which define exact location on the genome from which we intend to make multiple copies of that DNA. Oligos are sometimes also used. The term oligo is also used intermittently for the primers. Template DNA is that DNA for which we want to make multiple copies of DNA. That's called the template DNA. GC content in the PCR reaction is that defines about the pairing of the double strands of DNA, whether it's a strong pairing or it's a weak pairing. The more GC content we have, that means the DNA will be more strongly paired, and it will be harder to take apart the two strands of the DNA. Melting temperature is that temperature on which half of the primer is annealed to the strand, to the single stranded DNA, and half of the other primer will not be annealed. For example, if we have double stranded DNA and we denature the DNA into two strands, and we want to anneal temperature the primers, one will be the forward primer and the other will be the reverse primer. The Tm is exactly at that point where about half of this primer will be annealed to this template strand. Thermocycler is the machine which is used to carry out this polymerase chain reaction. There are some kinds of Taq polymerases which are high temperature resistant polymerases, like the Taq polymerase. So Taq polymerase is derived from Thermus aquaticus, which is basically a thermophile archaebacteria, and the DNA polymerase extracted from Taq is resistant for high temperatures. Then you might come across with this terminology of isothermal amplification and multiple strand-displacement amplification, in which the polymerization reaction occurs only at one temperature instead of using the thermal cycling conditions of annealing and extension and then denaturation, that the whole PCR can take place at only one temperature. And different enzymes used for these are like the Bst polymerases and other polymerases. So the basic purpose of today's seminar is to stress about the primers. So what are the primers which are used for polymerase chain reaction in PCR? If we make an analogy that if we want to make photocopies of certain pages from a book, then we have to define the page numbers that we want to make copies of this book from page number, for example, 10 to 20. Then it means that we have marked these pages that we only need multiple copies of these pages. So we're not going to copy other pages. So this is an analogy with the primers, that in using the primers in this reaction, we want to make copies of a particular piece of DNA, and that piece of DNA is marked by the primers, the forward and reverse primers. So for example, if this is forward primer and this is reverse primer, then this region will be copied, this particular region will be copied, and we won't get multiple copies of the regions beyond these boundaries. DNA polymerases are the enzymes which are used for making copies of these reactions. DNA polymerases need two types of sequences for the reaction. DNA polymerases are actually those enzymes which are used as the engines for making to add up these building blocks, and these are the polymerases. The work of polymerases is actually to add nucleotides into the growing nucleotide chain. So these DNA polymerases need two types of sequences. One type of sequence is this primer sequence, from which they will extend their sequences, and the other one is this template DNA, on which the primer will anneal. So binding sequence are duplex DNA, where the polymerase binds and starts amplification in 5 to 3 direction. And as we all know that DNA is DNA is basically composed of 2 strands, and these run in anti-parallel way. So if one has 5' end, on the other end the same strand will have a 3' end, and the template strand will have 5 to 3 that will run in anti-parallel reaction. So polymerization is always carried out from 5 to 3 directions. So DNA polymerase cannot go into reverse direction, that can only proceed in one direction, which is 5 to 3 direction. So for copying this strand, DNA polymerase can only move towards this direction, and for making another copy, DNA polymerase can only move from this direction to this direction. So this will be a 5 to 3 direction. And the template sequence are opposite strand that will be used as complementary. So DNA polymerase can add nucleotides, but for adding nucleotides it requires a template on which it will add the complementary nucleotides. So as we all know that there are 4 types of nucleotides, and A always tends to bond with T, and that has a double hydrogen bond, and G always tends to bond with C, which has a triple bond. So it needs this particular sequence as a template to add these nucleotides onto the growing polynucleotide chain. So basically it needs two types of sequences or DNAs. One is the primer sequence, and the other one is the template sequence. And if we see that this is a DNA amplification process, then this is a single-stranded DNA, and the primer, which is annealing on this single-stranded DNA. The primer is this particular sequence is the primer, and then DNA polymerases extends this single strand into a double strand like this, and it has reached up to this point at this moment. And these 2 strands are joined together by weak hydrogen bonding. So it's not a covalent bond, it's a hydrogen bond which can be broken down into single strands, and that can be re-annealed again to make double strands. So coming to this topic of choosing appropriate primers, we can pick up primers either from the literature, or we can de novo design new primers depending upon our requirements. So most of the cases we have in our lab, we have come across this situation in which taking the primers which are already reported in literature, that doesn't work well, and then we have to design new primers. So for my own, in my own experience, designing new primers, customized primers, is better than using already reported primers. And also in picking up primers from available literature, it's necessary to see that what type of primers are already reported in the literature and what has been their objective or use in the literature. So we need to pick up those primers which have been designed according to our own strategy, our own study. For example, if somebody is interested in deep phylogenetics, then it means that he or she will need primers which are complementary to stable regions, which will amplify the stable regions. But if somebody is interested in population genetics or investigating different populations or different individuals from one particular species, then it means that he or she will need those primers which are basically designed for the hotspot regions instead of for stable regions. So we need to take appropriate primers depending upon our own study, instead of using any primers which are reported in the literature. And these primers should be appropriate for the kind of study. For designing new primers, there are some general characteristics which we need to consider while designing the new primers, such as the objective of the study. For example, if our objective is to design primers to anneal to a specific DNA, piece of DNA or specific gene, that means that we need to design primers that have a certain length. But if our objective is to design primers for generic primers are low, are for those regions which are not specific for particular genes, but for generic primers, like we have been using randomly amplified polymorphic DNA markers like rapid markers, then those rapid markers are basically the primers which are lesser in size, so they have more chances of annealing to many different regions or multiple sites across the genome. So depending upon our need, for example, in most of the cases, we are interested to amplify particular pieces of DNA or particular genes. That it means that we need to design those primers which are specific for a particular piece of DNA. That means first thing which we need to consider is the size of primers. Okay, the size of the primers. So for annealing to a particular locus on DNA, the size of the primer should be between 17 to 23 nucleotides long. Depending upon different uses, we can custom design primers which are of longer range. For example, we can add non-specific tags or some tags onto this region. For example, if we want to pool different reactions for multiple other applications like in NGS, like in amplicon sequencing, we may need to add M13 tails towards the 5' end of these primers. So the length of the primer will be longer than 23 bases, but the region which is used for binding will be just between 17 to 23 nucleotides long, and that will be towards the 3' end of the primer, and this region will be used for annealing with the template DNA. The second thing is that we need to pick primers from those regions that are not complementary to each other. So this is to avoid primer dimers. And as we all know that the primers are also pieces of DNA. These are the oligonucleotide DNA, pieces of DNA. And as the DNA can make double strands depending upon the base composition, so in selecting primers, we need to select those regions which are not complementary to each other, so that the primer dimer formation could be minimized.
So can I ask a question?
Sure.
How to avoid this hairpin loop while you are designing a primer?
We need to pick up those conditions. We need to pick up those features while primer designing in software, that we can specify the size of primers, we can specify the length of the amplified region, we can amplify the melting temperature, we can specify the melting temperatures of the primers, we can specify the GC content of the primers, we can specify the dimer temperature, primer dimer temperature, and we can specify the temperature for hairpin formation. So for example, if some nucleotides within one primer are complementary to the other nucleotides, but then the overall annealing temperature of the primer is sufficiently higher, then that means we are again trying to open that hairpin formation. So for example, if this hairpin formation has been due to only 3 bases, that means if we elevate the temperature to a certain extent, then these bases which are complementary to each other, they will be in open conformation like this. The next thing is melting temperature, or TM, of the primers. And Melting temperature, as I have already defined, is that temperature on which half of the primer is in annealed form and the other half is in open conformation. And the melting temperature can be roughly estimated. It depends upon multiple parameters like the GC content as well as the other features, for example the primers, the medium of the primers and ionic strength of the primers. But generically speaking, there is a rough formula for calculation of the melting temperature, which is a rule of thumb, which is this one. The melting temperature can be inferred as 2 into A plus T plus 4 into G plus C. So for example, in this case we have a primer which has the size or length of 20 base pair, and all of those 20 nucleotides, let's say, are A and T, AT content. So what will be the Tm of that particular primer pair? 2 into 20, because all A and T are all the bases are A and T, and 2 into 20 is equal to 40 degrees centigrade. And what if we go to a hypothetical situation in which we say that all the 20 bases are G plus C? In that particular case, we need to multiply this 20 with 4 into G plus C. So that will come up to 80 degrees centigrade, which is a super high melting temperature for a primer. And that's the hypothetical case. The two are the hypothetical case. This one is roughly close to the normal or generic situation which we come across multiple times. For example, half of our primer length comprises of A and T and the other half comprises of G and C, and that in this situation 50% is G plus C and 50% is A plus T. The melting temperature will be close to 60 degrees centigrade, and this temperature is an optimal temperature. That means that for our annealing we can lower the annealing temperature to 57-58 degrees centigrade, which can easily amplify your desired piece of DNA. And if we have more G plus C content, that means we are adding complexity in the primers, and that will have difficulty in annealing to its particular temperature, because there has been recent studies which say that in those areas which have higher G plus C content, the double base pairing that turns into other structures of DNA, and that's one reason that there are multiple other structures which are formed in the DNA that basically hinder the annealing of primers to those regions. In our experience, the best temperature for annealing is around 55 degrees centigrade. So while picking up primer, while designing the primers, it's good to go for those primers which have Tm melting temperature around 55 to 60, within the range of 55 to 60 degrees centigrades. There are some other conditions which are important for the success of primers. So for example, quality and quantity of template DNA. The quality and quantity of template DNA is very crucial in successful amplification of PCR or polymerase chain reaction. The other important thing is that whether the DNA is dissolved in TE buffer or in double distilled water. This is important because TE buffer, when DNA is dissolved in TE buffer, TIS and EDTA buffer, EDTA is that particular agent which is used as chelating agent for magnesium ions, and this particular EDTA has dual functions. This EDTA chelates the magnesium ions, and magnesium ions basically are the cofactors for two types of enzymes which have opposite functions or different functions. For example, DNA polymerases also use magnesium as cofactors, as well as DNA endonucleases. DNA nucleases also use magnesium as cofactors. So it's important that when we extract DNA and when we have to store DNA for longer periods of time, we generally prefer to store DNA in TE buffer, because EDTA will chelate any available ions, magnesium ions, in the DNA solution, so that it is prevented from DNA is prevented from DNA degradation. Yes, but at the same time, for PCR, when we use that DNA as a template DNA, the EDTA which is present in the DNA will also be added into the PCR reaction, PCR mix, and that EDTA will actually chelate the magnesium ions. So we need to add certain amount of magnesium chloride which is higher than the chelating capacity of EDTA which is already available in the template DNA, and the extra magnesium which is available during the PCR reaction will be the extra magnesium ions will be used as cofactors for DNA polymerases for amplification reactions. So it's important to have appropriate concentration of magnesium chlorides. We need to work around the appropriate concentration of forward and reverse primers as well, and this is important because the forward and reverse primers are binding to the template DNA, and template DNA could be from multiple sources. For example, if our template DNA is coming from bacteria, from microbes, or from phages which have smaller genomes, then smaller concentration of primers will be enough to amplify to the multiple copies of that DNA which is already available in the template. Compared to those bigger template DNA, for example, if we want to amplify DNA from wheat genome, or from onion genome, or from human genome, which are sizes in the range of GBs, 3 GB, 6 GB to even 15 or 17 GBs of DNA. That means the total number of copies of that particular gene in the available DNA will be lesser. So we need to work around the appropriate concentration of the primers depending upon the type of DNA. Then we also need to consider about the concentration of dNTPs, which are the building blocks in the PCR reaction. Then there are usually there are two different types of buffers. One is the ammonium sulfate buffer and the other one is the KCl buffer. Ammonium sulfate buffer is normally used for making more stringent conditions, and KCl buffer or potassium chloride buffer is normally used for lesser stringent conditions. Then the important factor is the Taq polymerase itself. Taq polymerase, as I previously said, that it's the enzyme which is actually catalyzing the reaction, which is actually adding the nucleotides into the growing nucleotide chain. So depending upon the company of Taq DNA, the maker of Taq DNA polymerase, the concentrations of Taq DNA polymerase, the units of Taq DNA polymerase in the reaction, we need to figure out how much we need to have to add in the reaction. And then we need to work around the molarity of each of these components in PCR reaction, and for making these molarities we adjust the final volume from the double distilled water. And then after that, after making this mixture, we need to work on the annealing temperature or thermal cycling conditions for the PCR reaction. There are some differences in the primers between different types of PCRs. For example, conventional PCRs and real-time PCRs and next generation sequencing PCRs, for working on the RNA molecules, like we need to check the expression of certain molecules, or for depending upon the objective of your study, we need to design different primers. So for example, as I earlier told, that some primers are designed to anneal to multiple template sites on the template DNA. For example, rapid markers, RAPD markers or molecular markers, and ISSR markers. These are the markers which are designed to anneal to multiple different sites on the template DNA. And then there are other primers which are designed to anneal to specific single locus on the DNA. So depending upon these ones, we need to pick up appropriate primers. For amplifying using RNA, we can amplify using RNA, but then we need to convert that RNA into cDNA. So basically, when we have to quantitatively check the expression of genes, because gene expression is dependent upon tissues and time of expression and tissue of expression, we can quantify the expression, and then we have to start from the RNA. But instead of using directly RNA, we first need to convert that RNA into a single stranded cDNA for amplifications. At other times, we might need to pick up primers for amplicon sequencing and next generation sequencing. And in most of the cases for amplicon sequencing, we need to add some tags or barcodes at the ends of those primers. And for adding tags or barcodes, as we have different projects which are already going on, we have attached M13 tags towards the 5' end of these primers. So the length of the primer will be longer because of attachment of M13 tail, which is about 18 bases long. So the length of the primers might be from 38 to 40 nucleotides, but actually 20 of those nucleotides, or 18 to 20 or 23 bases, will anneal specifically to that DNA, and the other part will be added in successive reactions so that we can pool those amplicons for NGS reactions. There can be some influence of the primer design on the results. Designing of the primer is very crucial for the success of PCR. Normally there are three types of primers which can be synthesized. After designing the primers, normally we can design common desalted primers, we can order for page purified primers, and we can order for HPLC level purified primers. Well, the problem with the common desalted primers is that during the synthesis step of these primers, when we order for the synthesis of these primers to the commercial companies who synthesize these oligos, shorter chain oligos are already accumulated as byproducts. So for example, we ask them to design a primer which has the length of 20 bases, but then the short length byproducts of 19, 18, 17, 16, 15 bases and so on, these will also be added as a byproduct in the primers, and the actual concentration of your required primer will be variable, and these primers can have difficulty in annealing to specific temperatures, especially after certain periods of storage. And if you can avoid such primers, it's better, because the success of your PCR is more important. People can go for these common desalted primers because these are less expensive, but it's better to go for purified primers. Like Macrogen can also provide MOPC purified primers. These are also good for annealing to specific loci, or you can ask for page purified primers or HPLC grade purified primers instead of common desalted primers. There are some companies which are offering such services of designing, commercially designing and synthesizing the primers, like IDT from USA, Thermo Fisher US, Merck Sigma Aldrich, BGI from China, Macrogen from Korea. These are some companies which you can contact for designing the primers. You will send them sequences for particular sequences of your primers, and then they will synthesize these oligos and will ship you those synthesized oligos, and you will receive those synthesized oligos or primers in lyophilized form, and then you have to reconstitute those lyophilized vials and forms into your working concentration. So normally what we do, we mostly generate the master or 100 nanograms concentration of the primers, and then we prepare working concentration of primers that ranges from 5 nanogram to 5 nanomole to 10 nanomole of molarity. Troubleshooting in PCR, there are certain situations in which you are trying to amplify the regions, but at the end of the day, when you run the gel, you can't see any primers, so it means that there has been no amplification. Then you need to check whether there are some ingredients in your PCR which are inhibiting the PCR reaction. So for example, your magnesium ion concentration might be lower than available TE in the template DNA. And the other thing that can be important is that your primer is trying to anneal to those regions which have high G plus C content. So that's another reason, because as I told earlier, that high G plus C regions are those complex or complicated regions which are not easy to amplify. The other thing is that sometimes in reported literature, the sequences of the primers against certain genes are reported inaccurately. So for example, you have been trying to amplify such genes for multiple times, but there was no success of PCR, and when we checked that particular gene with the primers, we couldn't find any primer which could anneal to that template DNA. So that means that there has been some error or problem in reporting the sequence for that particular gene. That might not be belonging to that particular species either. There are times when just making the dilution of template DNA is sufficient for success of PCR reaction, but you might need to run the PCRs in a gradient mode. So for example, in one reaction you are going to pick up that best annealing temperature in one reaction, which you can use in your successive reactions. Sometimes there are situations in which we need to make a hard start for Taq polymerase, because Taq polymerase, we all know that it can work on high temperatures, but it doesn't mean that it cannot work at low temperatures. So when we are preparing the master mix at room temperature, sometimes the primer can start annealing to non-specific templates which are single-stranded, and Taq DNA polymerase starts amplifying those regions. Once it starts non-specific amplification, then it becomes difficult to direct it back to the specific regions. So in that situation, when we have to stop non-specific amplification at room temperatures, we might need to go for hard start PCR, in which we prepare our master mix on ice, on low temperature at ice, and then we start the thermocycler, and the temperature of the thermal block is 94 degree or 96 degree centigrade, and we directly transfer our PCR tubes or PCR plate from low temperature, from ice, directly to 96 degree centigrade for specific annealing of the primers to their particular respective loci. That's called the hot start PCR. Sometimes we also need to go for the touchdown PCR, and the touchdown PCR is that strategy in which the initial reactions start at higher annealing temperature, then we successively and gradually decrease the annealing temperature in our next reactions, so that at higher annealing temperature the primer will not anneal to any temperature, and the next reaction when we are decreasing the stringency or decreasing the melting temperature of the primers, we are basically directing the primers to anneal to the specific targets instead of going to non-specific amplifications. So these are called the touchdown PCR. This can also be used for avoiding non-specific amplifications. Normally we keep the lid temperature of the PCR about 10 degrees higher than the annealing temperature, so that when the thermal block is at 94 or 96 degrees centigrade, it's a sufficiently high temperature, and the water at that temperature tends to evaporate towards the lid of the PCR tube. And if the water is evaporated towards the lid, that means the concentration of reactants or reagents in the tube has changed. So how to avoid the water going towards the tip or the tip of the tube is basically you can increase the temperature at the top. So the lid temperature is, if that temperature is kept at about 10 degrees centigrade higher than the thermal block, then that basically suppresses the tendency of water to evaporate, and that the whole water remains inside the PCR reaction. There are some PCR stabilizers as well, which might need to be added from time to time, like betaine, trehalose sugars, and glycerol, and in some reactions we might need to add some mineral oil layers on top of the PCR tubes to reduce the evaporation of water from the PCR reactions. There are some software which are available to design the primers. The most commonly used is Primer 3 or Primer 3 Plus, which can be used online, and that Primer 3 has been incorporated in different paid software, for example in Genius as well. Genius also uses Primer 3 software, the plugin for designing the primers. You can also design primer on NCBI's tools like Primer Blast and IDT oligonucleotide primer, and there are some free available online resources on which you can design your primers. And it's important that while designing your primers, you need to consider the conditions which we have discussed earlier. Thank you very much.